Okay, so I still starting by update the toolbox. So today I will use the example I showed you in the lecture about the cell-cell communication. We are using real data. So what you can do, let me on the web page. I will use the example in the PowerPoint slide. So this session ID is what I'm looking for right now. We need to have this to download this example data. So in this case, I provide this link, this ID for you to get the data. In your research, you probably will have your own data set, or you want to download some data set from the reading the paper relevant to you. And also, you know how to download this, okay? Remember, if you have accession number and you check, they provide the essential, the matrix file, and you can just select this accession number and give that number in there. I'm quickly doing this because this data set is it's big data. So if you don't have this, you just type that in, okay? GSM 3121363. In the same time, you can also Google this ID and find this geodatabase record. This is from the mouse data and from the skin on the tissue cells. It's uploaded by this fellow and from UC Aaron, but they didn't publish anything, so we don't have any information about their annotation or their study, but looks like it's from this organization. Oh, they published this already. So the sample we downloaded from here. They do have this publication link to here too. So we can have a good reference because they already annotate for us. So when I download this data, I have no idea they published already. So I just blindly did the analysis myself. So we can have a cross-validation by looking at their paper and then our annotation, see whether we found the same thing. So after we process this data, then we will save it, and then we will look at how can we run single cell scTenifoldXct, which Yunjian present his package in MATLAB. But that one, we need to install Python. So it's a mixture. I will show you how to do this once we set it up, just like we set up for the R environment, then you can call R. But this package developed in Python, and I have an interface through this tool, but we have to install Python, that package separately. Okay, I want to make sure you get this, just like I have already. Take me several minutes. Then we need to use the filter because this is a huge data set. There are 22,000 genes and 27,000 genes, 22 cells. So we need to do a basic filter. What I did is stringent, and just use default, no whitelist. So this operation is also will take several minutes because the data is larger than normal. Normally we have 10,000 cells, 10,000 genes, but their data set is huge. So I want to make sure we are on the same page that we can move forward. We got this? Not yet? Have to wait because the computer is slow. Yeah. It will take forever. Yes, I know. Yeah. Yes, good. I just have to wait. Yeah. Good. So if you don't have an ID, you can check, okay, this feedback message just gave you where to download this. The program just did this automatically. So using this time, I just want to check this C-linker. This is a database for ligand receptor gene set. By the way, the information has already been built into the package, so you don't have to do this yourself, but you know where the information came from. It's just a list of those pairs, okay? Okay, the processing of my data is done, and now 13,000 genes have been removed because of low expression or other reasons for cutoff. And then we still have 22,000 cells. That's still a large data set. So I'm just going to do very quickly, because that will take another 5 minutes to do the embedding. Use t-SNE. So after that, I'm going to save my file and just in case. Perhaps just a routine. Okay, you should know this very well wherever you have a data set. To do this initial process, we need to know clusters, we need to know cell types. Especially in our case, we need to have cell types because we are going to detect interactions, right? So we can use this time to look the mini codon, which is a environment we are going to install for Python. Okay. Because we are waiting for the t-SNE to finish embedding, we can spend some time look at this mini codon package. Okay, by some click, you may come to this page at end where we will download this package, for example. So it's mini codon with Python, the latest version, basically. We are going to install this locally. to prepare your MATLAB and the Python environment side by side. So we can call the function. Yeah. Yeah, if you have Mac, you may, you should also max with this Python in at the same time. By the way, I'm not so sure. Yeah, it should work. It should work. So I have to test this in Macintosh machine, but in PC, I'm sure it will work. Well, my t-SNE is still running. I know it will take 5 minutes because the size of the data. In the same time, I can download this and install. So if you select just me, and then this will install it in your local profile. So you don't need administration power to do this. Okay, t-SNE embedding is finished on my machine. Also, the installation of mini codon is finished. So at this stage, I'm going to save this file because just in case the computer gets crashed because of memory use, I don't have to re-download the file. I can save this like single cell scTenifoldXct example. Okay, I saved my file. I feel safe right now. I can go back to check my installation of mini codon. So once you install that, you will have this command line based window. So you can click this and to get this shell. And then if you type Python, then we know it already installed. Okay, so we will establish communications between Python environment and MATLAB. Yeah, you don't have to do anything here. You don't have to learn those, what I will just exit. We just check, okay, we are ready to to use Python. So let me go around and look at if you have that. Okay. So different people may have different favor, use different package, but the mini codel is okay. Yeah, good. Good. So you have, yeah, you have Linux, yeah. Have a different, I don't know how to do it. Mac people, it's a different cat. Yeah. What you're doing here? Yeah. Oh, you can save anywhere. You can save to local. Yeah, just save. Yeah, you save. You give a new name. Yeah. Next time you can load it back. Yeah. Just put a place. I don't think this is a good place because yeah, maybe here this place will be good. Okay. So now I'm going to the page at Google, single cell scTenifoldXct. So the first link will lead you to the installation. So this is the let me see if we can do this. We need a Git. We need to install another two things for this environment. So pipe maybe not there. Okay, it's there. So pipe is a common. Git maybe not here. So I have to install Git. Let me consider how to install. So I will Google install Git for mini conda. Okay, we need this to let me try this. Okay. So in order to install single cell scTenifoldXct, we need this small command to install a so-called program called Git. Okay. So Git will help us to download single cell scTenifold. Say yes, that's all. Yeah. Yeah, it's here. Yeah, I will check. So I highlight what I type in is this. Yeah. Then you say yes. So we just need to use conda to install Git. That's it. I don't see what user. Oh, you have to come out. So that you are in the Python environment. So you come back in this place. You type that. Okay. Conda install. Yeah. Yeah. Yes. All right. Thank you. Yeah. So you need to exit from the Python. It's not Python. Then you start to type conda. Yeah. Yeah, so if you are still in Python, you have to, yeah, I think you are done. Now, if you have a Git, yeah, you are done. So we are ready to do next step, okay? Yeah, you are done, good. Yeah. Oh. So once you finish this, we can go to the single cell scTenifoldXct to install using single cell scTenifoldXct. So using this line of code. Okay. So this code just use Git to help you to download the package, and then use the pipe to install it in for your Python. That's all. Then we are ready. So I will just copy this, and once my Git is done, yeah, just 1 second. Go back to this page. Okay. Yeah, you copy this and put back to this black window. Oh, I cannot paste. So if this Yongjian made this, so if anything wrong, ask him. It's right there, okay? We are testing your package in different machine and also including a Mac. The reason Yongjian developed this in Python because within this pipeline, we need a neural network, and so very convenient for him to use this established other packages. The Python provide a lot of other third-party tools. Maybe sometime I can convert that into MATLAB completely, but not now. If I can make a MATLAB version, then we don't have to install this native MATLAB will handle this. But I'm not so sure the machine learning part, which, for example, from those packages, I can completely repeat, re-implement in MATLAB. So at this moment, we still use this combination of two languages. Okay. Are you ready? Finish this part. Good. So let me just repeat what we have already done so far. So we installed this minicodon environment. Minicodon support Python included in the package. Well, we also installed this Git, a small program to help us to download single-cell scTenifoldXct and use Yunjian provided the command line to install single-cell scTenifoldXct in this machine already. If you are a Python user, you can, so for example, import single-cell scTenifoldXct. If this comment give me nothing, so that indicates it's already working. Okay, you can in Python this work. We are not going to deal with this because you have to learn Python how to read the matrix in. So we are going to use MATLAB to call this. Is that okay? If you reach to this step, that means you have successfully installed the package and you are ready. We are ready to go. I believe for your real data analysis, it's worth it because single cell communication is a big deal to get something out from your real data set. That's something that we cannot do with traditional bulk RNA-seq. So it's adding values to this analysis. So we spend some time to set this up. Yeah. No, you have to be in Python. THON. Now you can do the import. You get it. So you have to be either in the base or in the Python environment. Yeah. XCT. Yeah. No, let me see. Did you install this? So you haven't finished the installation.
Oh, okay.
Yeah, it takes some time to run. So
Okay, so
yeah. So we have to get out from this environment and to run this again. So this, yeah, this will take time. Yeah. Yeah, once this completely finished, we should good. Yeah. So if you have already installed this successfully, we can switch this off. We don't need this interface at all. So I will do exit the Python and then exit this environment, minicodon. Come back to our MATLAB. So now we are going to annotate the cell. So first do this over clustering. So I will cluster them into 100 small clusters. And then use alt cell type annotation to automatically this is mouse data and select mouse to that annotation to finish job and merge if we have a lot of fibroblasts in there. So we will finish the annotation and take a break, come back to for the next step. You have to be patient because this data set is larger, the bigger than normally you should get. I don't know how they get 20,000 cells. This is mouse data, yeah. 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 QC filter including mitochondria cutoff. Yeah, by default. Yeah, I set up a relaxed and stringent. Stringent is not so stringent at all. You can change it a little bit. So I want to keep a lot of gene and the cells in general. Yeah, but that QC, the manual should do, normally should do the work. Yeah. Other several steps. Yeah. So if you want to really careful about those, you can use several steps manually. It's just basic. Yeah, very basic. So once it's finished, I'm going to merge those subclusters with the same name. There are 5 different groups. And I probably will combine this pancreatic stellar cells with hepatic stem cells, stellar cells. So what I'm going to do, have you got this page? Okay. So what I'm going to do, I just rename them all to stellar cell instead of. So I can click this button to rename cell type. I select pancreatic stellar cell to stellar cell. And rename hepatic also into stellar cells. So if they have the same name, these 2 should be joined as the same group. Now we have only 4 groups. Let me just relabel them by the color. Okay, at this stage, I'm going to save that file again to just before the break, okay? Okay, at this stage, I want to look at their original publication, see what cell type they annotated from there. And they use cell chat to do this communication. What we can do is to find this geodatabase record and then find the link from the original study. Reference. In this paper, so they review fibroblast heterogeneity. Yes, we certainly have a lot of fibroblasts. This in this blue, and some looks more similar to stellate cell. Some is, yeah, so there are subgroups in there. So it makes sense. They also said they have adipocytes progenitors. We don't have this annotation. Let's see what they have got in there. Okay, normally the figure 1 will contain this t-SNE plot. Let's see, they have T cell, B cell, a lot of, where's the T cell? So we have a C1, 2, 4. This 1 is here, 2 is here, 4 is here. So this 3 groups are the fibroblasts. I think these 3 groups are what we have here. The blue one. So they make 3 subclusters. We have endothelium cell, which should be this purple. Purple in this group should be what we have here. And then this light blue myeloid cells is what we annotated as stellate cells. That's what I'm thinking. Then they have some T cells, Schwann cell. So they have red blood cell. Let me check red blood cells. So you see this tail color, this small group. Red blood cell has very unique marker of the gene. So in this manual, we do have this special show this gene expression. Okay, so this will help you identify where blood cell is, where we do have some. So looks like there's small clusters here. Maybe it's this one. Have a combination of those markers of blood cells highly expressed. So I don't know exactly which group. This is. So maybe one of this group is blood cell. Okay. Then they have some immune cell annotation, which some of these small clusters, they don't have macrophages. Yeah, so maybe you can check some of their fibroblast, myeloid cells, gene expressions. Those are marker genes, they believe this group, CD68. Which group? Somewhere here. They are B cells, but I believe they are macrophages. So that's a marker for the endothelium cell. And that's for fibroblasts. So they have their reason to believe some clusters are T cells, some are B cells. I'm not so sure. For example, I can check CD3D, and they believe there are some cells in this center are T cells, but let me see. CD3D in our expression profile. See. Yeah, it's not so clear to me where the T cell are distributed. They're not in the cluster. Right, maybe it's here we have a small cluster, they believe the T cells. Right, so this really depends on your understanding of your system and then which marker you really trust. Myeloid, like CD68. I know CD68 is macrophages marker genes. CD68. It should express highly in this group. Yeah, so this corner here. Well, this also, the whole cluster has high expression of this. The whole cluster is it's really hard to maneuver this. Okay, it's this group here. This macrophage. Okay, so there are some annotation they inconsistent between our annotation with theirs. I think we can use multiple markers and to solve the problem. I'm not saying we are wrong, but maybe they didn't check enough markers to give assignment for this. Is macrophages called myeloid cells, original from the myeloids. Any questions with this part? Fine. I think they only show one marker gene. They probably just use one marker gene. Yeah, but for illustration purpose, they just pick the top one. They believe it's strong evidence. But of course, we shouldn't just use one marker. We should use more. Okay, now let's check the cell ligand receptor pairs we used as an example. The particular one I use is this pair, okay? CAV1 versus PDGFR A or B. There are two receptors, either A or B. So what we are going to do is to show those genes expression profile. So what we can do is just select 3 genes. One is CAV1. This is the ligand expression, and the receptor PDGFR. PDGFR A and B, 2 genes. So this is the same receptor with the 2 subunits. And look at their expression individually, one by one, 3 genes. So we should have 3 this plot here. So let's see first. Or if you want to rotate these 2 subfigures at the same time, you have to click this link button here to make them synchronized. You click this and they become synchronized. So the purpose of this is we have to look at this gene, which cell type highly express that gene. So it's this group, right? This group is, in our annotation, is this yellow group, endothelium cell. Okay. So CAV1 highly expressed in endothelium cell, we annotated in this cluster, this group, also shown in this bar plot. And its receptor, let's see where the receptor highly expressed. Oh, it's into the fibroblast. So if you look at the receptor B, it's hard to see. Yeah, it's difficult to see, but my conclusion was it's highly expressed in the stellate cells. Okay. So that's by visualization, we create some kind of this link between endothelium cell highly expressed CAV1 and this receptor was highly expressed relatively in stellate cell. So that's why this communication relationship was established. The receptor A is not so clear. Receptor A is highly expressed in the fibroblast. Okay, so we probably can draw endothelium cell produces ligand and give to fibroblasts, and the fibroblast uses subunit A to receive the message. Well, it gave to stellate cell use subunit B more to receive this message. I don't know, but this is like a computer inference to try to help others. So now the question is why I look at these pairs instead of in the database, there are thousands of pairs. This, of course, is through detection of the significant LR pairs using XCT, for example. You can use many others. Let's check what they use CellChat to find in the original paper. So they have this original paper to infer analysis, cell-cell communications, use cell chat. Let's see mouse data. The reason I look at this paper because they mention in their record, which probably this study used that data set. So we have to find it out. So I'm going to use this ID to see whether it has been mentioned in this two development paper. Okay, single cell data, they use this test data. This is in the section of methods. So this group of authors use their own data, which is this one. Okay, I have to search their results. It is skin cell during, so I believe figure 2 is relevant to what we are, the data set is we are using, and they did this analysis that's across different cell types because you have a lot of subgroups of fibroblasts. They did this individually across their targets. So what they present here are the pathway level. So they not present like individual ligand receptor pairs, but they group them functional together. They report like the whole pathway. TGFB signaling pathway may involve 5 different ligand receptor pairs. So they try to build this relationship between the whole pathway and the two between different cell types. Okay. So all these are not the name of genes. It's pathway, signaling pathway. Each pathway may have 5, may have 10, may have 20 pairs of ligand receptor. But on statistics, if you as a group, you are significant, they will include that in here. So this design is slightly different from what we are doing. So what I see from across the whole section, they only look at this pathway level of enrichment or significance. The next section, they use different data set. Okay, so let's see what we can do. Okay, what we need to do, for example, we are going to run single cell scT. Please don't run this on this data set because there's too many genes, the huge matrix. If you build a network 14,000 by 14,000, it will blow up your computer. It's such a big, so we need to have some condense. We need to just pick the most important genes. Like 7,000 is the maximum I can try on local machine, 7,000 genes. So we have to find a way to throw away half of the gene in this data set before we can actually run. But for the demonstration purpose, I will only use 500 genes, a small network. Okay. So what we need to do is first to use external to check our Python environment, because right now the link between them has not been established. If I check, we need to find this where the Python has been installed. Okay. Has not been set up, so we need to find where the Python is. Are you with me at this step? Okay, we are going to click and try to find where Python is. So I know it's installed in, let's guess, C user. I found my name here. In the column. Oh, wrong place. Let me go back where I installed not here. I'm trying to find where I installed the mini codon. Inside the download? No. Download? No, not this one. Let me see where I installed the mini codon. Okay, I can see, yeah. Okay. Okay, I know. It's in C user and found your name. There's I will search. Okay, so that's where the location is. Okay, we don't have that. So probably I will copy this folder. Yeah, it's pretty hard for me to locate where this is. Yeah, at the end, I think I found it where this Python is. So I can copy the location. So wrong one. But I will type in the doc here. So notepad. Yeah, so the Python should locate in this folder. So it depends on your username. But in some system, this app data is not visible. But on my machine, my office, normally you should have this visible. But on this machine, I don't know why. Your profile and your app data has been hidden and become a system level. But I'm going to try this. Identify where this Python comment is located. I think the backslash. I will just type in exe. Okay. Oh, not supported. So I won't be able to show you because the version I'm using on this machine, my MATLAB, is a little bit old. Okay. My MATLAB version is older than the Python version. Python is 10.3, so I cannot connect this with a higher version of that Python. So if I want to use this locally, I have to go back to mini column, but download the older version of Python. So you have Python 7.4. But if you have a new version of this MATLAB that support the latest Python version, so it doesn't matter. I don't know what's your version on your machine. It's a same problem. Okay. But which part? Yes, there's an error message. It's just show you the incomparable.
So versus
oh, you shouldn't. You let's see. Let's shut this down and then yeah, close. You should type the whole thing in here. Yeah, yeah. Okay. .exe.
Oh,
if yeah, I can.
Okay.
Not valid. So.
Okay.
Oh, I think this is like just backslash instead of slash.
Oh, okay.
Yeah, then you should be okay. But that's just a way to set up. We only running into this incomparability problem. Let me show you how to do it.
Yes, sir.
Yeah. So you have a C folder, user, your name. So you just change this back to for the Windows machine.
Oh, okay. Versus it's okay.
Yeah. Local. Yeah, so this should work. So that's a way you just add that. You can copy this just in case. Yeah, so this should work. Should add that in, but because not support. So you get this problem. Yeah, yeah. So the rest of time I will go back and download. You are right. Everything is right. I will download this older version of Python, and I will very quickly redo installation for the. Okay, so I will do the mini cordon conda. Yeah. So let me take a look. So we cannot use. I will see 3.8. This will be safe. This should be safe. Maybe even earlier. 3.7. I'm going to install 3.7. So we know where we installed. Maybe I can change this to somewhere I can really find. So I just change it to here. So next time I know where I installed. I just installed under my personal profile folder. Okay, I installed Python 7.3, 3.7. So now I can come back to here to see, I check my Python environment and locate. Now I know where I installed. So I know it's under user, my name, mini codon, and Python. Yeah, so now I think I'm ready. But in this package, I haven't installed scTenifoldXct, so I have to redo the scTenifoldXct. Have you get this step here? You reach this step? No, I need to know whether this is which version this Python is. Okay, that's good. I need to install conda, install Git. So I confirm that my current Python version is 3.7 and it's connected with my MATLAB version 2001. I install Git and also this through a pipe. Okay. I just repeat what I did for the latest version of Python. I downgrade to the version that I can use in this machine. Oh, why? Shouldn't be too hard. Oh, you have to exit from Python. Yeah, exit out. Yeah, so from here you install Git. Yes. Right. Yeah, Git is just for download from GitHub. Small stuff. Yeah. Okay, good. Yeah, change it. Otherwise, you cannot find it.
Yeah, unless you just copy and paste, copy that.
Yeah, yeah. Well, yeah, that's good.
Okay, yeah, I guess it'll be right under your phone.
Yeah, that's good. So I just check my installation. That's in this version of Python. And then if I do import single cell scTenifoldXct. Okay, it's working. So I can switch this off. Come back to here. It's ready to use. So this is already set up, so I know this is correct. Suitable. Now we need to make a small data set. We need to do this top. Just select top highly variable G, and then you put 7,000. Then it's supposed to be able to run on your local machine. Otherwise, the network just oversized. So you select this and put 7,000 here, you should be okay. But for the demonstration, I will only use 500. It's a very small network now. Use this method, and then the number of cells will be the same. We only select those highly variable genes for our downstream to make it smaller. I choose for the neck. Yeah. we will discuss highly variable genes and their biological meaning significance after the spring break. But this is a good way you can sample your genes, but keep the most important genes. The highly variable genes are the most important genes. So my matrix is much smaller right now. I can, okay, right? Much smaller. So when you let me know when you finish, we move forward. We are at this stage. So if you finish this step, you can click the network, and then at the end, you select this cell-cell communication. I've put 3 turtles here. It's really because it's slow, it's real. So it permit you again and permit to see whether you are using this version of Python yes uses. And now you have to select cell type. So we will do this too. Select cell. And then once you finish, you click done, then you will see, okay, you want to study interaction from endothelium cell to stellate or back or both direction. Yeah, to this step, you have to click the network, then cell communication interaction. Make sure we have a small data set, 500 genes, okay? Otherwise, we won't be able to finish today. Cell communications use this Python, and then cell type or not ID. Yeah. 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 Input, yeah, just give a test. Have the trouble, you haven't installed that package. So you need to go back to the GitHub page and to, yeah, copy this. You already installed? No, let's go down a little bit. Are you sure it's finished? Okay. Maybe just try that one more time. Yeah, yeah. Number of cells, yeah, so that's in the, let me just click this and let the program continue. And you'll see cancel. Okay, so right now the program is building first network for your source cell type, and then build another network, and then to do the manifold alignment. Yeah, so it's here, the experiment. Then you select, oh, you use older version of the MATLAB. Yeah, so your version is older than what I have. Yeah, yeah.
But I downloaded that one.
I don't think so. You downloaded today. So that should be if you download today. Yes, this is older version, so you probably have to reinstall this package by overwrite it. Yeah. After that, this place allow you to select not 2000, but you can say 500. Yeah, yeah. Okay. But for now, you can select 2000. Let's see. We'll only finish 2000. There's too many. There's too many. Yeah.
What is this actually?
We call it highly variable genes. Highly variable genes. Yeah, we will discuss next. Yeah, yeah, yeah. You need a new version of this package. So on my screen, it's running the step 3, which is a manifold alignment. So I already constructed 2 networks for two cell types, and then try to align them right now. Yeah, git say yes. Yeah. Yeah. So when we use only 500 genes, some ligand receptor gene will be missed because they're not included. Yeah. A lot of errors. Yeah. Yeah. Yeah, that's by default. That's fine. Yes. I don't know. Which cell type? You can select if you have 3 cell types, you can select first 2, 1, 3, 3, 2. You can you decide yourself. Oh, let's select endothelium cell and the stellate cells. Okay. Yeah. That's for example. Did you save the file? Yeah, it's good. Yeah. Oh, from beginning, that will take long. Did you save the file in the middle? No. Okay. Oh, by the way, you can use just example data. Yeah, just use example data. We will have the same. Yeah. Just load the example data, processed. And yeah. Yeah, so no, you can select the top like 500 here. Yeah. That's right. Then I don't know if you have set up this. I don't know. Yeah. Right. Cell type. Yeah, you select 2 cell types you want to play with. It doesn't matter which one, which 2. Yeah. Yeah, that's true. So you can use example data. So cell type. Yeah, you select 2 cell types you want to study the interaction between.
I selected, but the problem is it's
yeah, then done. Yeah, so yeah, you haven't installed this package from here, not I think from yeah, you haven't installed this because if I do Python, yeah, this is correct version. So that's a problem. So you haven't installed that correctly. Yeah, so you have to install it. Okay, you did this. That's good. Then did you copy that pipe? This
pipeline.
Yeah, the pipe. If you go to yeah, you copy this. You have to finish this command. Then you are done. Yes. Yeah, you have to wait until this finish. Because this only install git and cannot. weird. Where I don't know. No, I think you still have the problem of the Git. You didn't install the Git. So you have to install this correctly. Finish this, then install that. Then you go back to MATLAB. So you say yes, yeah. This takes time. Yeah. Okay. So once you've finished, we should have a table which have a pairs of these significant ones. So the top one I found is this interleukin 6 against a notch 3. So I can come back to here and to see by profile interleukin 6 versus notch 3. Look at their profiles. Yeah, because we only include 500 genes, so this top ones maybe not really. Okay, so but that's just a small demonstration. But when you use 7,000 genes, some significance should jump out. You got the direction. This, for example, this is from a endothelium cell to stellar cell, and one gave the ligand and the receptor. Okay, and then at the bottom here you have the different direction from stellar cell to endothelium cell. That's by the algorithm. Okay. Yeah, that's all. Okay, so for real data, prepare a computer with a big memory, and just wait until something happens. Yeah.
Is this injection data also considered as p-value?
You don't have to consider p-value. You only consider rank. If you think top 50 is meaningful, you use top 50. If you think only 15 of them is you want to report, you include 15. It's just a rank there. Yeah, you can set up a cutoff anywhere you want. Yeah. Maybe not in the list. Oh, yeah. Yeah, in that case, you want to keep that gene. So yeah, you can do something like this. You can see, okay, I want to keep top 8,000 highly variable genes, see whether the gene you include is still there. If not there, you see, okay, I want to keep 9,000 genes, it should be there, right? Then you manually delete some genes, make this slow, not delete the genes, yeah. That's yeah, I should give the function say I select the white list, no matter how I won't delete that gene. So I can provide some function like that. Yeah. I think for the local machine, yes. Otherwise you have to set up this environment in the cluster. Clusters you can have much larger. networks, genes. Yeah.
Not only we also have a biological connection.
Yeah. Yeah.
Based on it's not random.
It's not random. It's based on the database. Yeah, only from the database.
How can we visualize? Make an example visualize data.
Visualize, you mean you visualize what? You can select these two genes and to do this visualization, just I show you in the, yeah, so to this bar plot, the profile plot. But sometimes you just don't see the signal because it detects as average somehow, but not visually appealing. Sometimes it's convincing by looking at them. That's some good examples. Yeah, but some of these are very weak link, but because it's supported by the network, they push them together. Yeah. So we have tested this at least in real study, at least in one of my collaborators, they found it's really good. Yeah.
This point is why
it's a distance, the smaller. So this gives you some idea about how close they are. Yeah. So okay, the distance is here. The correspondence is something else. So maybe you can ask Yongjian what he put it in there. So the distance should be the rank the same. Yeah. The correspondence maybe is this interaction row score. Yongjian knows. I don't know what he put in there. Yeah, try this with some with your hypothesis. Yeah. You can try with like 2,000 genes first, then expand to 5,000 genes, and at the end 7,000 genes. Because once you have the list, you have some time to think about it. Then you expand your space of the genes. Yeah.
To put it into a network.
Oh, you see a network, and yeah, we have to find a way to how to plot the network. It's hard. Once you have a large number of genes, how can you draw the network? It's a big problem. Yeah. I think so. Yeah, it's not exactly this database, but the one we can have the most comprehensive one. Yeah. It should be, yeah, should be in there. Yeah. Maybe I should give a function that allow you to set up your own database. Say I want this maybe your own customize the database. I will set it up. Oh, I cannot because that's in Python. So you just have to provide this function. Yeah. What I have here is just an interface allow you to give the cells to the Python program and come back with this table. So what the Python is doing over there, Yongjian control it. Yeah. Okay. Yeah.
So at this step, if you've already annotated all the clusters with their own cell type, would there be a difference between the cluster ID and cell type?
That will be different. The cluster ID will be original if you separate it into 100 small clusters. Cell type is by name. Yeah. Do you have cell type? You should have. You can just click OK. Yeah, you have 4 cell types. Yeah.
Some problem in installation.
Did you install this? Yeah, so I don't know. Yeah.
Okay.
Yeah. So that's basically this is missing a function. So this function is supposed to be in one of this folder. Yeah. Yeah, maybe just override, maybe just reinstall this. You shouldn't have that problem because everything has been tested. Yeah, inside here, you should have the geo function. So it's not there, right? But if you check your version, you should have that in there. So, yeah. Yeah. Reduce the number of genes. Yeah. So this in the experiment.
Okay.
Yeah, there are top n highly variable genes.
Oh, okay.
Yeah, you can see like 500. Yeah.
Okay, great. No,
Yeah.
presumably that for some done on
That's take a long. Yeah, that will take some time. 500 will be just a couple minutes.
Okay.
500 should be fine. 500 genes definitely. Yeah, yeah. Definitely should work. So next week will be spring break, no class. Next week, the Tuesday class, I have to cancel it because I have a meeting travel. But the Thursday, we will come back to this room. So next week, Thursday, come back to this room. We will talk about the highly variable gene, and I will give a lecture. In the same time, we can play with. So we combine basically the computer session with the lecture. Okay. Okay, I do have a recording for this. Okay, if you miss anything, and there's a link.